Thank you very much for this kind introduction and for giving me this opportunity to discuss our research. So it's really a pleasure and honor to be part of this prestigious lecture series. So today, let me actually use the other pointer. So I need to move the screen away. Apologies. So today I'm going to discuss about 3D imaging of flow in porous materials. So most of my talk is related to rocks, but later part of the presentation, I'll show how we can use the tools developed for subsurface research in exploring other research avenues, for example, termite nests or ant plant symbiosis. So for imaging, we use medical CT scanner, which is very similar to what you see in hospitals like this one here. So here what you can do, you can put an object on the table and you can throw X-rays on one side and then capture on the other side. It gives you a 2D image. And then you can rotate the whole assembly at different angles to get many different 2D images, and then you can combine them together to get a 3D image. So we use a similar technology in laboratories. The difference is that instead of moving the whole assembly around an object, we move the object or samples on a rotation stage. And another difference is that we get higher resolution. Here we can get down to hundreds of micrometer in pixel size, but here we can go down to 1 micrometer, which is sufficient to resolve pore spaces in rocks. Generally, so this is a typical image we can obtain from these micro-CT machines. This is a Caton limestone. This is like a 5 millimeter in diameter sample and imaged at 5 micrometer voxel size. So in this case, we took multiple images and then stitched them together to get a complete sample. We can also do local tomography. That means we can zoom into and get higher resolution images to resolve the pore spaces, tiny pore spaces. So here what we have is like black is the air in this case because it's a dry sample. And then this is the rock grains, which is in gray. So in our experimental setup, we put this sample in a core holder, which is almost transparent to X-rays. So another thing is that it can withstand high pressure, high temperature conditions, which can mimic reservoir conditions. So generally we conduct these experiments like two-phase flow, multi-phase flow experiments, which has importance in many different topics, as you may know. So first is like in CO2 sequestration, we are doing a couple of projects. Here you inject CO2 to displace, let's say in saline aquifer, you're displacing water. Although there are many other geochemical processes happening, but today we'll focus only on two-phase flow problems. And then for oil recovery, when you inject seawater to displace oil from reservoir rocks. Here the complexity comes in terms of wettability of the rock space. Another examples where we find multi-phase flow problems is soil remediation or how the contamination occurs, and many other natural industry processes where we can find these multi-phase flow problems. So one of the factors which can influence these multi-phase flow problems is the wettability of the rocks of the porous media, which is characterized by the contact angle at the three-phase contact line. Conventionally, traditionally it's measured through the denser phase, like if you got air or water system. So if the contact angle is less than 90, we call them wetting surfaces, otherwise they are non-wetting surfaces. So in this talk, what I will present, how the wettability can influence two-phase flow in porous media. For most of my experiment, what I'm going to show today, we use a synchrotron facility. In this particular case, it was diamond light source. The reason for using it is the speed, imaging speed. For example, if we get a 3D image in a lab for within 2 or 3 hours, here we can get the same image in 2 or 3 seconds or few seconds. That means we can resolve the pore spaces during flow, so we can quantify, visualize the dynamics of multi-phase flow in porous media. So we'll start with the very simple case, water wet. It's a Caton limestone. It comes from a village called Caton in the UK. Here, what you see is a pore space, and these are the rock grains here. It has microporosity. It has some complexity in this. So with the experiment, we fill this initially with water, and then we inject a non-wetting phase. In this case, we use oil to displace this water. This process, which is called drainage, you can see we see big jumps here and there. So this is a typical characteristics of a drainage process, which is mostly controlled by throat sizes. When the drainage is complete, when there's no, let's say here I only show oil, water is not shown, also rock is not shown. But when the drainage is complete, when there's no flushing of water from the rock sample, so we reverse the process. We inject water from the base, and this displaces the oil in the porous media. So here you can see the processes are quite different. We don't see big jumps like what we saw in drainage. These are like more smooth processes. So it's mostly controlled by pores, but also the throats play a role when you have the snap-up processes, which we'll discuss later. Another thing is not a reversible process. As you can see, there's lots of trapping after once the water flooding is complete. So this is the final image after imbibition, obtained after slightly more than 1 pore volume injection of water. So different colors here, they show disconnected components of oil. We call them ganglia. People can call them droplets of CO2 if the CO2 is there. So they remain stable because of capillary forces if we use the same flow rate. If we can increase the flow rate, they can be displaced, or we can use surfactant, soap water to displace them. I put one picture to show our efforts decades ago to characterize these ganglia. These techniques were mostly destructive those days, but nowadays we have got a luxury of using tomography to image and visualize and quantify non-destructively. Now, because we have this time-resolved imaging from Synchrotron facility, we can also zoom into various locations and can see how these oil droplets or ganglia getting trapped. So here in this example, you see oil and water is not shown here. And you can see as the water pressure is increasing, or if you keep injecting water, we get a pinch off or snap off leading to trapping in this pore. We can also zoom into this throat and can see how the water is actually expanding there. So this blue is water. With the time, as we increase the water pressure, this water layers are swelling and leading to trapping when we see the pressure disequilibrium. So you see the throat is completely full of water after we get the disconnection. One thing what we can do in terms of analysis, we can convert the interfaces in 3D to curvature maps. It's a water oil curvature maps, which we can convert to capillary pressure using simple Young-Laplace equation. So in this small subset, what we did, just to give an example of the analysis we performed here. So what we did, we first divided this whole image into 2 parts. Left, we call them like ganglia side, and the next one is on the right-hand side is connected. So here what we plot capillary pressure calculated from the Young-Laplace equation using curvature maps as a function of time or injected pore volume. And here what you see red is the left side, which is ganglion side, and black is the other side. So as you increase the water pressure or the capillary pressure is decreasing with time, so both pressures are decreasing at the same time. But then a point comes when we start to see the disparity in pressure. So the pressure on this side starts to decrease and it keeps decreasing. And then what it allows, it pushes the brine layer into the throat. So it allows for throat swelling or brine layer swelling in the throat until we get instability where we see the snap off here. I'll discuss slight bit of instability at later part, but this is just to give you an example of what sort of analysis we can produce. Here, what another thing is that the brine layer swelling, the speed here and the time it takes is between 10 and 15 minutes, which is quite slow compared to what you see for a roof snap off, which is in milliseconds when you have drainage process. Another thing what we can do, we can convert the pore space into pore-throat network. Here what you see are spheres, they are pore spaces, and these tubes, they represent the throat radius. It's just the representation in terms of tube or a cylinder connecting different pores. Now, because we got this time-resolved imaging data, we can put our oil on top of it. And again, water is not shown here. We can see how the interfaces are moving. It's like a pore filling event. And then we get snap off and trapping here. So with this sort of analysis, we can characterize the pore and throat space, radius, shapes, and many other parameters. We can see where the pore filling occurs and where we get the trapping. So this is an example analysis in terms of what we found in correlations. So let's say, just to give you an example of one sort of analysis, let's say this is our fluid configuration here. So we call it like I1 fluid configuration because one throat is filled with oil here. But here we call it I3 because 3 throats are filled with oil. In this case, it's a non-wetting phase here. So for this particular configuration, what can happen? Either when we increase the water pressure, either it can move, or before it moves, the wetting layers, they are swelling, like I shown in 3D, they can lead to snap off and trapping of this. And similarly, in I2 events, you can have snap off or I2 pore filling events here. So what we did, we looked into what's the relationship or dependence of these events. Either it's going to have snap off or pore filling, dependence on the pore space geometry. For example, pore radius, throat shape factors, coordination numbers means how many throats are connected. And the last one is aspect ratio. Let's say fluid moves from here to there. So the aspect ratio is the radius of this pore to the radius of this throat. Now, what we did this correlation analysis on the whole data set. If we got 0 value, that means it's not dependent, not correlated. But if it's -1 or 1, it has some dependence or correlation with the pore through geometry. If you see all the parameters, this aspect ratio quickly outshines, and it's really clearly is correlated very well, not fully, but very well. To the pore throat spaces. So this analysis actually we can, maybe in the future, we can use in pore network models to improve them. But again, some more work is needed and also more experiments are needed to generalize this aspect. The minimum aspect ratio we found from these experiments is slightly more than 2, which is consistent with the theoretical values, but is slightly more than what people have found on 2D micro models. So maybe the differences are the nature of 2D and 3D there. During the same sort of experiments, we found some unexpected events. For example, this is the fluid configuration during imbibition. This is the oil, and this is an I1 event because there's only one throat connected to this oil. Now, when you increase the water, and then what's happening? The interface moves to the next pore, but we don't see sort of conventional snap-up process in throat. Otherwise, we would see a trapped oil in the center of the pore space, but rather we see something sticking to the pore wall. So we were curious. Sorry, I think I've got this message on front of my screen about the host. Give me 1 second. Let me remove this. Apologies for that. So we were curious how actually this sort of trapping can occur. So when we looked into grayscale images, this is real images from the experiment, and this is oil and this is brine. When we see the contact angle, it looks slightly more than 90. Rather, we have got a water wet system. We start to see some sort of angle which is not expected. So it can occur under different conditions. Let's say if we got some surface active compound, like in crude oil, it can change the wettability. But in this case, we had a very simple decan. So we won't expect this sort of wettability alteration there. Second thing could be minerals. If you have different minerals, it can lead to interspinning or different contact angle. But we had a 99.9% of calcite surfaces in this particular rock. Just to clarify, this white thing shouldn't be considered as like a different mineral. It's just an imaging artifact. We start to see a phase contrast in the imaging here. The third possibility is if you got rough surfaces, interface while moving, it can pin and can give us a larger contact angle. So this is our hypothesis because there's no other two possibilities here. Unfortunately, we can't really resolve this roughness because of the resolution, but hopefully in the future with nanoscale imaging, we would be able to answer this. And this is the image after trapping. One might say, okay, this is because if you see real, it looks like a pore pocket. But when we saw carefully in 3D, it's like a expanding pore. We don't see throat in between. Even if it was a different pore, we would expect trapping somewhere inside the pore, not bulging outside here. So we were curious if we can use some simulation work, because experimentally, we got this limitation on time resolve images. We can only see before and after. And Musaib Shams from Imperial College, he performed very nice simulations for us. So what we did, we used contact angle we obtained from images with 45 degree from the whole sample. It didn't really trap there. And what we did then, we knew the trapping occurs here. And we said, okay, although we can't resolve the roughness, but we know the contact angle changes. Maybe we can make a patch and we give a different contact angle to this patch, and then rest 45 degree. And then we gave 60 to here and 40 to 45 to other places. We didn't see any trapping there. But as soon as we made it 70, and we see the trapping, what we saw in experiments here. And if we increase the contact angle, we still see the trapping. It's only because of contact angle we see different sizes. So it's dependent on the contact angle we put in the simulation. And the second thing, the size of this trapped ganglion is dependent on the patch, the size of patch we use. We actually wanted to go further, like why this thing happens. So what we did, we took multiple images, snapshot from simulations, and then because as I showed, like we can do curvature mapping and calculate capillary pressure. So we did the same analysis here, and then plotted the capillary pressures versus time here. So what we see initially the pressure is slightly dropping, which is expected, but then at some stage we get some sort of instability. The pressure quickly starts to rise here. So this point is somewhere here when you start to see transfer curvature, the interfaces move the other way around, and this radius start to shrink and shrink. That's why we're getting the higher capillary pressure. And then after some time, then we get the trapping here, snap off here. So this instability is somewhat similar to what you could see in a conventional traditional snap off, where you can have snap off in a throat. But the difference here is this sort of snap off doesn't occur in throat. It's rather happening in the pore space, which we related to surface roughness. But again, we need more research on this topic, maybe with some 3D printed micro models with careful selection of roughness or wettability or something like that. But I think there's quite a lot of work we can do in this topic here. Okay, this was all about water wet system, but we did quite a lot of experiments on wettability. So for this, we did very simple experiments. We took a sample filled with oil and spherical particles, monodisperse of same size, and then we injected brine from the base and captured the dynamics with the very fast speed 1 to 2 seconds at the ESRF in France. And then we repeated these experiments by systematically changing the wettability of the whole system from 20 degree to 160 degree contact angle. So these are two examples of 75 and 125. What you see on the left side here, or both sides, they are cross-section, vertical cross-section through the sample, to the center of the sample, middle of the sample. And what you see here, black is oil, initial oil, and these grays are the spherical particles. And what we do now, we inject brine from the base in both contact angles, and we can clearly see a very compact front. And here we got more ramified or finger formation in the front, dependent on the contact angle. And this is similar in 3D. What you see here, different colors, they show different time steps of water injection. Only water is shown, but different time steps. And 75 degree, 125, we see a compact front. And here we see quite a lot of ramification of the front or the finger formation. We did some sort of different types of analysis. One was very obvious. We did front analysis, but obvious was like because we see ramification of the front here, we expect to see a larger area, interfacial area. That's what we see here in the plots when plotted as a function of injected pore volumes or as a function of time. And in this case, we don't see much like, because it's a very flat front here. Another thing which is expected, that because it's a flat front, we get to the residual saturation quite soon with one pore volume. And also the residual saturation will be low because it's very flat front. We don't see ramification. And this is what exactly is oil saturation versus injected pore volume. And then we get to the residual saturation for this case within one pore volume. So these things were expected, but we just quantified to show what differences we see this with the contact angles during flood. But the highlight, I would say, is this plot here. When we plot 3D residual saturation from these images as a function of contact angle. So we see almost no trapping in these cases. And then suddenly we get quite a lot of high trapping for these particular cases here. And we also compared these experiment with 2D experiments on the micro model from our colleagues. Although the values are different, but the shape of the transition is very similar. And the point to note here is the transition doesn't occur exactly at 90 degrees. So this is like it's a range from 90 to 120. So somewhere here the transition occurs for this particular porous media. And that means when, for example, when we go more than 90, we move to drainage state, from imbibition to drainage. And in drainage, we always expect we're going to have big bursts like Haynes jump. But in this range, we might not expect bursts in jump, but rather other instabilities like touch or overlap. Touch means when the interface moves, it sees another obstacle before jumping, or two interfaces can merge together before any jump occurs. So this sort of finding, actually, they are related to sphere packs, very monodispersed packs, homogeneous porous media. But what we've seen, the wettability can alter quite a lot of things here: residual saturation and the front area or the shape of the front. Another thing, what we wanted, if we keep the contact angle same, and then change the system, make it more heterogeneous. For example, here we mixed two different types of beads, sizes of beads. And although we see sort of compact front, we see thick fingers forming in ahead of the main front. And similarly, if we go to more heterogeneous system, sand, rounded sand, or with sharp corners, we see, rather than getting fully displaced, we see quite a lot of trapping there. So we can imagine, like, one axis we got wettability, and the second we got heterogeneity in the porous system, which can control the fluid displacement and trapping. I think the interesting part would be if we can do this sort of experiment, what I showed for the glass bead or beads previously on the rock sample. But the challenge here is how to really systematically control the wettability from 20 degree or to, let's say, 160 degree. I think some work needs to be done so that we can also change the contact angle and the heterogeneity in the porous system. And the last bit before I move on to the other topic is the mixed wet system, which are more related to oil industry. It's believed the oils formed in the source rock and then moved to current reservoirs. So the reservoirs, initially they are water wet, and the oil, when it moves as a non-wetting phase. But over time, because of, let's say, surface active compounds or asphalting, the surfaces can change the wettability, can offer a mixed wet state. So we did quite a lot of experiments with crude oil and also with the synthetic oil. So this example is for a synthetic oil. What we did, we injected oil, started drainage, and this is what you see, only the oil. Water is not shown, rock is not shown here. And in this oil, we have, like, we added static acid, and what it did, it changed the wettability, allowed to change the wettability overnight. But if you use the crude oil, it takes really few weeks. We've done quite a lot of experiments with that as well. So it changes the wettability overnight, and then we inject water to see how the oil actually looks like there. So this is the oil after flushing about 40 pore volumes. of water. The structure is quite different what you find in a water wet system. One thing is the residual saturation here is about half, but the interfacial area is 3 times than this one here. So we wanted to explore it further. So we zoomed into to look into few pore spaces, what's happening, how these thin plate-like structures are forming there. So this is the image after drainage. The larger pore is filled with oil, and then corner we can see water sitting there. And overnight, the wettability changes. And when we inject water, it behaves like a non-wetting phase, and it displaces as much as oil it can do under that pressure conditions and leaves behind oil in the corners, or it could be sandwiched between two water bodies here. So these are like oil layers, which are like very different from what we see in a water wet system, where oil is sitting nicely, relaxedly into the larger pore bodies. So difference again, residual saturation can be achieved in this system within one pore volume. And by here, if we keep injecting water, these layers slowly drain and can produce more oil. But the other thing is that you need quite a lot of water to displace or drain these layers, which might not be economical for industry. We saw this sort of layers also in three-phase flow when we injected gas along with that. But today's presentation is only focused on two-phase flow here. Okay, just wanted to point out that the data set what I've shown, most of the data sets are available, especially the water wet case what I've shown initially. All the images are available online on public domain. And if you like to do more analysis, or if you like to compare it with your simulations, please feel free to download it, or you can send me an email if you have difficulties in downloading them. Okay, so we'll now move on to another topic, which is also porous media, and we use again 3D imaging to explore some of the biological system this time. You might have seen this sort of structures in television or in real. They can grow from 2 meters, 1 meter, 2 meters to 7 meter high. The fascinating thing about these structures are they are made by termites who are blind. Even though they're blind, they can make really complex structures like these ones. These are like casting images from inside what we have in the nest here. So for decades, biologists were interested in exploring these nests. The motivation, let's say, this is a cartoon of the nest. And if we measure the temperature outside, depending on the country, it could fluctuate from 15 to 30 degrees centigrade day and night cycle. But inside, not underground, inside the nest, the temperature stays within plus minus 2 or 3 degrees centigrade. So it's really well controlled temperature condition in the nest. So the people have done studies if you got abandoned nests, if you don't have termites, comparing both, we still see the temperatures is maintained very well. So it comes because of the structure of the nest. And if the motivation here is, if we understand the mechanisms of this temperature control, we might have a possibility to design energy efficient buildings. The second thing people have thought, this is blocked. Nothing can pass through, which didn't make sense to us because there are millions of termites living there. And the carbon dioxide they produce, it needs to be ventilated. It needs to be exchanged with the atmosphere. So this nest they have to be porous in nature. Now, there are two different types of termite, like nests you can find very large ones where you can have large pores. The termites, they open and close these holes for depending on wind velocity, for ventilation, but they can't keep it open permanently because they can be attacked by ants like eating them. So they close and block depending on the condition. For smaller nests, they don't have those sort of openings, and then somehow the ventilation has to occur through these walls. So with this motivation about learning ventilation and thermoregulation, we made some nice collaborations with the French researchers who gave us access to nests from Africa, from Senegal and from Guinea. And these are the nests sitting there. And then they brought into the laboratory and we scanned them using a medical CT scanner at low resolution of the order of 1 micrometer. And what you see here, red, is the solid structure. And the yellow is where termites, they move around. So we're doing actually quite a lot of analysis. It's really at the preliminary stage of the structure of the nest. But what we did for this work, we were interested in looking into the microstructure of the wall. So we took subsamples from the nest and also made sub plugs out of this of about 5 millimeter in diameter, 5 to 10 millimeter in diameter. And then we put into our X-ray micro CT machine, and the images look like these ones here. This is 2D image from Senegal and Guinea, and this is same for Senegal 3D and for Guinea here. What we see here, black is air, it's empty space, and the gray or white is the solid space here. The one thing is very clear for when we saw these images, we got very small pore spaces here, and then we got very big spaces here. And interesting part was that these big spaces, they were connected from out to inside the walls. We were actually first curious how they might form this sort of structure. So we started looking into the videos. So what termites they do, they go around, collect the soil, put their saliva in their mouth and compact this soil. It's called pellet. It could look like this. And the compacted mass might have the pore structure, which is like very compact like this. But when you put different pellets together, up and down, the pore space in between could be big. Like if you pack spheres in a glass, you will have space in between spheres. And this is a large space. The shape of this will be dictated by the size or shape of these pellets and how they are packed. But that's interesting. And although it turned out to be, it's not intentionally made, it comes as a geometrical, like packing geometry problem. But we were curious if there's any benefit of these larger pores to the nest. So we did quite a lot of simulations on thermoregulation, ventilation, and it turned out to be the larger pores, if the wind is flowing outside the nest, they easily help the nest to ventilate. But it also turned out to be if there's no wind flowing outside, then the diffusion kicks in. And this can allow for passive ventilation, passive ventilation of the nest, exchange of CO2 to the atmosphere, which is really good for them. So it doesn't depend really on the wind flow outside the nest. Another thing you can see, because if the large pores, they're empty and it works as an air insulation. So it can give some sort of thermal instability to the nest. But again, we need quite a lot of work into the thermoregulation. One more thing, what we did, we thought, okay, let's assume it rains. And when it rains, what we did very roughly, we sprinkled lots of water on the termite nest and it soaked in all the water. Then it was clear that it's a water wet system. And then we made some small plugs and then we injected quite a lot of water on it. The white you see is water. And because it's a water wet system, it soaks the water, sucked in by smaller pores because of capillary forces. And the imaging, what it showed us, the larger pores, they were empty. Perhaps because we need more water pressure to invade these pores, but the rain, because it's just hitting the outside of the termites, it's not sufficient to create that pressure. But these pores remain empty is really useful for them. Let's say when the rain stops, the nest can immediately start ventilating. So it's really useful feature for them. So all these results we put together, and we were happy that we managed to publish this work. Now in the future, what we're working on various aspects, structural components, and also combining simulation at different scales. So our aim is to learn how actually they control the temperature, and then we can design hopefully energy efficient buildings. The buildings, they don't need to look like termite nest. It's just the idea is to learn the mechanism and design something which is livable. Another thing which we also working is if we can design based on the mechanisms some sort of future bricks or the stones for building blocks based on the microstructure of the nest. So this is again work in progress because these days we got 3D printing quite advanced. Perhaps with the learning from the termites, we can design some sort of new bricks. We were happy that our study actually got a lot of media attention. And although it was a really hobby project we used to do on the weekends, but we were happy that it got attention. But also we were more happy when we did public events. People were very curious about that because it's really easy to relate public have seen this sort of things in real or on television, and they were curious how these blind termites can make structures which can lead humans to design based on their designs. So this is our team, and the rest of the team is actually missing from the picture. This is my last slide. We also started exploring and plant symbiosis. Again, we're using digital rock physics to explain some of the biological questions here. The problem here is there are some ants in country like Fiji. What they do, they go around and hunt for seeds, and they are interested only in 5 to 6 types of seeds. And when they find that seed, they go high up, find a crack on a tree, and they put the seed there, and the plant comes out. And generally plants, they have smaller stems and then leaves. But these ants, they know that stem is going to be large like this, or even like this. And they give nutrition to this plant, and they protect from enemies. And in exchange, plant gives them habitat to live. So this is like a centimeter scale opening inside this plant. If you cut it and image it, cut it, and you can see these sort of openings there, and they are connected. When you measure the temperature is very stable there. And it's very similar to what you see in termite nests. So we are doing some sort of simulation to understand in a similar way. But here we got one more feature to consider is the plant is moist, and with evaporation, because of the latent heat, it can cool down. But I think we are still progressing with this research. It's led by Guillaume from Sheffield University. With this, I would like to wrap up. Like the message here is like with the 4D advances in 4D imaging, especially with synchrotron imaging. 4D means time resolved 3D imaging, has allowed actually us to investigate pore-scale displacement processes. There are many groups around the world who are working on this topic, and people have developed really nice tools to characterize wettability, capillary pressure measurements, and many other more parameters. And also we can check what's the effect on flow-wettability, but there are a few challenges how we can really control the wettability of the rocks so that we can come up with some sort of phase diagram there. But another thing, what the message here is the tools what we have developed for subsurface research or digital rock physics, we can use to explore other biological systems. We can help them. For example, what I've shown you today in termite nests and plant symbiosis, although we are just at the initial phases of ant plant symbiosis, but there is opportunity. And I think there are many more which I'm not aware of, but maybe slowly with time we'll get to know where we can use these digital rock technologies to explain some of the basic fundamental processes which can be used, for example, for human buildings or human needs. With this, I would like to acknowledge our sponsors for this project. At Imperial College, it was sponsored by QCC SRC. At Max Planck Institute, it was British Petroleum. And I'd like to acknowledge many people on different topics who have supported this work, really difficult experiments with synchrotron imaging, and also especially technical stuff, because it's a really complex experiment with the high pressure, high temperature conditions, and they have helped tirelessly to get to this point here. With this, I'd like to thank you for your patience and your interest in listening to my talk. I'm happy to answer any questions.
Yeah, thank you